Welcome to the Creative Commons RX podcast. Before diving into this episode, I want to ensure we're all on the same page. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.
Welcome back to The Deep Dive, the place where we turn complex, cutting-edge science into crucial knowledge you can use. Today, we are peering inside, well, the most personal instruction manual available, your DNA.
That's right.
Our topic is applied pharmacogenomics, the definitive deep dive into translating your unique genetic variations into clear, actionable clinical decisions.
It's a really exciting field.
It absolutely is. And our mission today is ambitious but fast. We need to quickly and thoroughly grasp the molecular foundations that, dictate drug response. We need to understand the sophisticated bioinformatics tools that interpret this raw, complex data.
Yeah, the digital toolkit.
Exactly. Look at real-world clinical applications for everything from common pills to revolutionary cell and gene therapies, and finally, confront the rigorous regulatory and ethical hurdles slowing the adoption of this technology. This is your ultimate shortcut to understanding how your genetic blueprint dictates personalized medicine.
And that roadmap is vital because pharmacogenomics, it's fundamentally about optimization and prediction. Our overriding goal here is to use this genetic insight to maximize drug efficacy and, just as crucially, minimize adverse reactions by tailoring therapeutic strategies to a patient's precise genetic makeup. We're discussing a paradigm shift that moves way beyond simple trial and error dose adjustments. In fact, for many advanced therapies today, a genetic test isn't just a guide. It is a mandatory prerequisite. It's like the ticket required for eligibility.
A gatekeeper, essentially.
Okay, let's unpack this molecular foundation then. If we zoom right into the cell, what is the core mechanism, the fundamental link between our genes and how we process medicine?
Yeah.
We had to start with the central dogma, right?
Yeah.
But framed specifically for this context.
Exactly. The central dogma RNA makes protein. That's the starting line for the causal chain in pharmacogenomics. Your genome, it's the structural blueprint for all the proteins involved in a drug's life cycle. Think about the metabolic enzymes that break it down.
The transporters that move it across cell membranes, and even the final drug targets themselves. If there's a variation in that DNA blueprint, well, it inevitably leads to an altered protein structure, and that affects its functional capabilities.
And those functional changes are where the clinical impact lies.
Precisely. The clinical relevance hinges entirely on that functional consequence. So if a variation results in, say, a slow functioning or even a non-functional enzyme, the drug is going to accumulate in the body, potentially reaching toxic concentrations.
Okay, that makes sense.
Conversely, if a variation results in an ultra-rapidly functioning enzyme, maybe it's synthesizing too much protein, or the protein itself is just overactive, it clears the drug way too quickly. This leads to subtherapeutic levels, meaning the treatment fails.
So you get either toxicity or failure, depending on the variant.
Exactly. That fundamental pathway from the DNA variation through the protein function, that's the mechanism that explains the staggering inter-individual variability we see in drug responses, across patients who receive the exact same dose.
Okay. So if the DNA is the blueprint, we need a complete lexicon of how that blueprint can actually be altered. We need to differentiate between the three main types of genetic variation: SNPs, indels, and CNVs. Let's break those down.
Absolutely. Let's start with the most common and smallest variations, the single nucleotide polymorphisms, or SNPs. This is where a single base, A, T, C, or G is swapped for another. SNPs are categorized by their impact on the resulting protein.
Right.
A standard synonymous SNP, or a silent SNP, occurs in the coding region, but doesn't actually change the amino acid sequence. This is because of the redundancy in the genetic code, where multiple codons can code for the same amino acid.
But I've heard that silent doesn't necessarily mean harmless, right? That term can be a bit misleading.
Oh, it's entirely misleading, yes. While it doesn't change the protein sequence itself, a silent SNP can still profoundly affect the protein's final abundance or even its shape. Well, for example, changing the codon affects something called the local codon usage bias. This impacts how quickly the corresponding transfer RNA, the tRNA, is recruited during translation.
Oh, okay.
This can subtly alter the speed of translation, which might lead to slight misfolding or even protein aggregation. More dramatically, a SNP, even if it's synonymous, could accidentally introduce or destroy an exonic splicing enhancer site, an ESE.
And that would mess up the splicing?
Completely.
Hmm?
It could lead to altered or skipped mRNA splicing, and boom, you get a completely different protein product than intended. So silent isn't really silent in terms of consequence.
Got it. So what about the SNPs that do change the protein?
Right, those are the non-synonymous SNPs, and they're often the ones we worry about most in clinical settings. These split into two main types. First, the missense SNPs, where the amino acid is substituted for another, like the I359L change we'll talk about later. And second, the generally catastrophic nonsense SNPs. These introduce a premature stop codon. The ribosome basically just halts translation early, resulting in a severely truncated and almost always non-functional protein.
Right, that sounds pretty bad for the cell. Okay, so moving beyond just single base changes, let's look at structural changes involving groups of bases, the indels, insertions, and deletions. You mentioned the reading frame is critical here.
Correct. Absolutely critical. When indels occur, the key distinction is between in-frame and frame shift. Remember, the genetic code is read in triplets of bases, these codons. If you insert or delete a number of bases that's a multiple of 3, like 3, 6, or 9 bases, you're essentially adding or removing one or more whole amino acids. This is an in-frame indel. The resulting protein might still function, perhaps just with slightly altered activity.
But a frame shift is
Totally different, and usually much worse. A frame shift indel involves inserting or deleting a number of bases that is not a multiple of 3, say 1, 2, 4, or 5 bases. This completely disrupts the triplet reading frame from that point onwards.
So everything downstream is garbled?
Exactly. Every subsequent amino acid is scrambled. And this almost invariably leads to the introduction of a premature stop codon shortly thereafter, resulting, again, in a severely truncated, non-functional protein. Frame shifts are essentially biological disasters for that particular gene.
Wow. Okay. And finally, we have the really large-scale structural changes, the copy number variations, or CNVs. These are needed to explain some of those massive gain-of-function phenotypes we see, right?
Precisely. CNVs are large duplications or deletions of entire segments of DNA, sometimes spanning whole genes or even regulatory regions. A gene deletion, like the well-known DFYPTD65 allele, result in a total loss of function for that entire gene copy.
So if you inherit 2 deletions,
Then you're a poor metabolizer. You have no functional enzyme from that gene. If you inherit 2 functional alleles, you're a normal metabolizer. Simple as that for deletions.
but the duplications are where it gets interesting for gain-of-function.
Yes, the really profound clinical impact often comes from gene duplications or multiplications. These are the primary mechanism for dramatic gain-of-function. If a patient carries, say, 3, 4, or sometimes even up to 13 copies of a functional gene,
13 copies, wow.
yeah, it happens. They end up synthesizing significantly more active protein than a standard patient. This overwhelming capacity for metabolism is what defines the ultra-rapid metabolizer, or phenotype. A poor metabolizer might have an activity score of 0, but a could have a score of 2.5, 3.0, or even higher.
Okay, so we've got this huge array of potential variations, thousands of SNPs, indels, CNVs, and they combine in complex ways. How on earth does the field manage this incredible complexity and distill it down into something simple and functional for a clinician who just needs to prescribe a drug today?
That leads us directly to the crucial star allele nomenclature system. You're right, the raw complexity of the genome is just too much for routine clinical practice. We have to simplify. The reason we can simplify is largely due to a concept called linkage disequilibrium, or LD. Basically, LD means that individual variations that are physically close together on a chromosome don't get shuffled independently during inheritance. They tend to travel together as a large, stable block of DNA. This co-inherited block is called a haplotype.
Ah, so variants stick together in blocks.
Exactly. And the overall functional effect is determined by the combination of all these individual variants within that block, that haplotype. The star allele system provides a functional shorthand for that complex haplotype. We assign a unique star designation, like 1, 2, 3, to a specific defined haplotype.
So, like, 1 is the standard.
Generally, yes. 1 is typically the reference wild-type allele considered to have full function. Then you have others, like 5, which often represents that non-functional whole gene deletion we mentioned, and those duplications causing the ultra-rapid phenotype. They're indicated with an XN suffix, like CYP2DTD61XN, where N tells you the number of functional copies on that allele.
That structure makes sense. So the ultimate piece of information that goes from the lab report to the patient's electronic health record isn't the star allele itself, but the combination of the two they inherited, one from each parent.
Precisely. That combination is the diplotype. It represents the 2 star alleles inherited. For example, a patient might be CYP2C913. This diplotype represents the total potential enzymatic capacity of the patient for that specific gene.
Okay.
Then, based on established guidelines from consortia like CPIC, and often by calculating an overall activity score based on the known function of each star allele, this diplotype is used to predict the patient's clinical phenotype, like normal metabolizer, intermediate metabolizer, poor metabolizer, and so on. This standardized functional language is really the only way to make genomics actionable in real-time clinical practice.
Right. It bridges the genetic complexity to a clinical recommendation. Okay, this is where the intellectual challenge becomes really practical. Once we sequence a patient, we just have this long list of A's, T's, C's, and G's, maybe some insertion codes. How do scientists actually translate that raw genetic data into something clinically meaningful? We need to look at the 3 critical databases that form our digital toolkit. Let's use the reduced function CYP2C93 allele as our case study.
Yeah, this bioinformatics process is really the engine of applied pharmacogenomics. We can trace the specific missense SNP RS10579110, which is the defining variant for the CYP2C93 allele, specifically the C.1075AC change, through 3 distinct levels of analysis: population frequency, protein function mapping, and ultimately its structural consequence.
Let's start with step 1, DBSNP. This is the essential archive, you said. But why is the first piece of clinical information we need from DBSNP the population frequency? That seems maybe counterintuitive.
It's a great question. DBSNP, the database of short genetic variations, is the primary public repository. It confirms the variant exists, gives us its genomic coordinates, basic stuff. But its most critical clinical contribution, often overlooked, is indeed the population frequency data.
How so?
If we search for our variant, RS1057910, DBSNP confirms its position, but crucially, it breaks down the frequency of the C allele that's the one associated with reduced CYP229 function across different global populations.
And what does it show for this one?
We find it's highly variable. The C allele frequency is around 11.4% in South Asian populations, drops to about 6.6% in non-Finnish Europeans, but it's only about 1.3% in African or African American populations. That's a massive difference.
Wow, 1.3% versus 11.4%. That is striking. Why does that massive difference matter clinically beyond just being an interesting academic point?
Because this allele frequency data serves as a direct proxy for the pretest probability. Think about a drug like warfarin. It has a narrow therapeutic index, meaning the dose needs to be just right, and it's metabolized significantly by CYP2C9. The cost-benefit calculation for preemptive genotyping shifts dramatically based on this ethnic background data. If you're considering testing a South Asian patient before starting warfarin, you know there's an 11.4% chance to carry this high-risk allele. That makes the test potentially highly cost-effective and clinically useful for preventing adverse events.
Okay.
Conversely, in a patient population where the allele is very rare, like 1.3%, the clinical yield of testing every single person is low. While you never dismiss the potential risk for an individual, the high cost of universal screening for a rare allele might not be economically justified from a public health system perspective. So the data from DBSNP helps guide resource allocation and informs individualized risk assessment before you even prescribe the drug. It also raises important questions about equitable access to testing across different populations.
That makes a lot of sense. Okay, so DBSNP tells us where the variant is and how common it is in different groups. Now for step 2, UniProt. How does this resource bridge the raw sequence data from DBSNP to actual protein function?
All right, UniProt is the comprehensive curated resource for protein sequence, structure, and functional information. We take our variant, RS105791DR, and we map it onto the specific human protein it affects, in this case, CYP2C9, which has the UniProt accession number P11712.
And UniProt does the translation.
Exactly. UniProt performs the translation from the DNA change, C.1075AC, to the protein change. It tells us this SMP corresponds precisely to the amino acid change PIL359Lu, often written as I359L. Isoleucine becomes leucine at position 359.
Ah, so that connects the dots to the protein level.
Yes, and the real strength of Uniprot lies in its expert curation. It doesn't just list the change, it often links that specific amino acid substitution directly to its known functional consequences. In this case, Uniprot notes the I359L changes association with the CYP2C93 allele and explicitly mentions the impaired metabolism of key drugs like warfarin and phenytoin, which have narrow therapeutic windows. So Uniprot is that crucial academic bridge translating a raw sequence archive like DBSNP into a clinically relevant functional summary for specific protein.
Fantastic. This brings us to the really cool part, the mechanistic insight found in Step 3, the Protein Data Bank, or PDB. The PDB lets us actually visualize this change in 3 dimensions. When structural biologists looked at the CYP2C9 protein structure containing this I359L substitution, I think the PDB ID is 5x24. What was the key, maybe surprising, finding about why this change impairs the enzyme's function?
Yeah, the PDB is the global archive for experimentally determined 3D protein structures. And the visualization here is essential because it immediately challenges the most obvious assumption. If you locate residue 359 in the 3D structure, you find it situated quite far outside the central active site. The active site is that hydrophobic pocket where the drug substrate actually binds and where the chemical reaction, the catalysis, occurs. A basic, naive analysis might look at that location and assume the mutation is benign because, well, it's not right there near the action.
But structure dictates function, as they say, and clearly that's not what happens here. If the change isn't directly in the active site, how does it still cause such a massive reduction in function?
That's the beautiful insight from structural biology. The analysis of the structure, like PDB ID 5S24, reveals that the I359L substitution, even though it's distant from the active center, induces long-range conformational shifts throughout the protein.
Okay, what does that mean practically?
Imagine you're trying to open a complex safe. The I359L substitution is like changing one single crucial screw on the outside of the safe door. It doesn't directly touch the internal combination tumbler,
Right.
but that small external change subtly warps the alignment of the entire door mechanism, maybe distorting the tiny access channel the key needs to slide through, or slightly changing the shape of the internal chamber where the tumblers sit.
Ah, so it's an indirect effect.
Exactly. This is a classic allosteric effect. The change in shape, caused by the subtle difference between the amino acid isoleucine and leucine at position 359, propagates through the delicate protein structure. It disrupts the precise geometry of either the substrate binding pocket itself, or, perhaps more likely here, the access channel that the drug needs to travel through to even reach the catalytic site. This impairs the enzyme's ability to either bind the substrate effectively or undergo the catalytic cycle efficiently. It's a critical lesson. A variant's functional impact is often determined by its influence on the overall protein shape and dynamics, not just its proximity to the catalytic center.
That's fascinating. So we have the variant, we know the protein it affects, and we understand the molecular mechanism, in this case, the subtle but crucial allosteric effect. Now let's move from the genome to the bedside. What does this all mean for the patient's actual drug response? We need to talk about drug disposition, the whole ADME process, and the resulting metabolizer phenotypes.
Right. The transition from the genetic information, the diplotype like CYP2D614, for example, to the clinical phenotype is usually formalized by assigning an activity score to each inherited allele. This score system allows us to categorize patients into roughly 4 main phenotypes.
Okay, what are they?
You have the poor metabolizer, PM, who typically has an activity score of 0, meaning virtually no enzyme activity from that gene. Then the intermediate metabolizer, IM, with reduced activity, maybe a score of 0.5 or sometimes 1.0, depending on the gene and alleles. Next is the normal metabolizer, NM, considered the baseline with fully functional activity, often scoring between 1.0 and 2.0. And finally, the ultra-rapid metabolizer, who has significantly increased activity, scoring above 2.0, often due to those gene duplications we discussed.
And the classic example gene for this is CYP2D6.
CYP2D6 is the paradigm, absolutely. It's responsible for metabolizing something like 25% of all commonly prescribed drugs, think SSRIs, beta blockers, opioids, many others. Classic examples of non-functional CYP2D6 alleles that lead to a PM phenotype include CYP2D64, which is a splice site mutation preventing proper protein assembly, and CYP2D65, the total gene deletion. If you inherit 2 of those, your score is 0.
But the clinical consequence, whether that patient risks toxicity or treatment failure, hinges entirely on the type of drug being metabolized. This is the crucial dichotomy, right? Is it an active drug or is it a prodrug?
This distinction is everything. It dictates the entire risk profile. Let's start with an active drug. These are drugs that are biologically active in the form they're administered. Metabolism serves primarily to inactivate them and prepare them for excretion. Think of the tricyclic antidepressant amitriptyline or certain beta blockers like metoprolol. Now, in a poor metabolizer, PM, the lack of enzyme activity means clearance is incredibly slow. The active drug accumulates rapidly in the body, leading to significantly elevated steady-state plasma concentrations, sometimes 5 to 10 times higher than intended for a standard dose.
And the consequence?
The clinical consequence is a very high risk of toxicity and dose-dependent side effects. For amitriptyline, this could mean severe anticholinergic effects, sedation, or even dangerous cardiac arrhythmias. Therefore, the PM usually requires a significant dose reduction, sometimes 50% or more, or switching to a different drug altogether.
And if that same patient taking that same active drug amitriptyline happens to be an ultra-rapid metabolizer instead, how does that flip the outcome?
It completely flips it. In a UM, the enzyme activity is massively increased, usually due to those gene duplications like CYPTD61XN. The active drug is cleared almost as fast as it's absorbed.
So it doesn't stick around long enough.
Exactly. This results in consistently subtherapeutic plasma levels. The patient might feel like they're taking a placebo, leading to treatment failure or a complete lack of response. The clinical strategy here is often complex. You might try dramatically increasing the dose, potentially needing 2 or 3 times the standard starting dose, but that carries its own risks. Often, the better approach is to switch to an alternative drug that isn't primarily metabolized by CYP2D6.
Okay, that makes perfect sense for active drugs. Conversely, for a prodrug, a drug that's inactive until it's processed by the enzyme, the risks are completely inverted. The classic example here is the opioid codeine, isn't it?
Codeine is the textbook example. Codeine itself provides very little pain relief. It must be metabolically converted by CYP2D6 into its potent active metabolite, morphine, to exert its analgesic effect.
So what happens in a poor metabolizer?
In a poor metabolizer, PM, this crucial conversion step doesn't happen efficiently or at all. The patient takes the codeine, but they experience a total lack of efficacy. The drug is simply ineffective for them, and they remain in pain. They might even be wrongly labeled as drug-seeking because the standard dose isn't working.
And the ultra-rapid metabolizer? This sounds dangerous.
It can be extremely dangerous.
Yeah.
The ultra-rapid metabolizer faces a high risk of toxicity. They rapidly convert even a standard dose of codeine into unusually high, potentially overwhelming levels of morphine very quickly. This can cause severe side effects, most worryingly, potentially fatal respiratory depression. You might remember the FDA issued black box warnings about codeine use in children who are UMs, particularly after tonsillectomy due to reported deaths.
Wow.
Another really key prodrug example is tamoxifen, used in breast cancer treatment. It needs CYP2D6 to convert it to its highly active metabolite, endoxifen. Poor metabolizer women often have significantly poorer breast cancer survival outcomes because they don't generate enough active drug, leading to reduced efficacy.
That prodrug-active drug dichotomy is crystal clear now and absolutely critical for translating the genotype. We should also touch briefly on phase 2 enzymes. These are the conjugating enzymes using the example of UGT1A1.
Yes, phase 2 enzymes are equally important. The UGT1A1 gene encodes an enzyme that performs glucuronidation, a process that attaches a glucuronic acid molecule to drugs or metabolites, making them water-soluble and easier to excrete. It's essential for inactivating various compounds, notably SN38.
And SN38 is?
SN38 is the highly toxic active metabolite of the chemotherapy drug irinotecan, which is used for colorectal cancer, among others. The key functional variant in UGT1A1 is called UGT1A128. Interestingly, this variant isn't in the coding sequence itself, but in the promoter region, the regulatory sequence just upstream of the gene that controls how much enzyme is made.
So it affects production levels.
Exactly. This UGT1A128 variant involves a variation in the number of TA repeats in a specific part of the promoter. People with the 28 allele have 7 TA repeats instead of the normal 6. This seemingly small structural change reduces the efficiency of gene transcription, leading to lower production of the UGT1A1 enzyme.
Okay, and what's the clinical consequence for an oncologist prescribing irinotecan to a patient who has 2 copies of this 28 allele?
Patients who are homozygous for this reduced function allele, 2828, have significantly impaired capacity to inactivate SN38 via glucuronidation. Because they can't clear this toxic metabolite efficiently, it builds up. These patients are at a roughly 50% higher risk of experiencing life-threatening, grade 3 or 4 neutropenia, a dangerous drop in infection-fighting white blood cells, and also severe debilitating diarrhea.
So testing is standard now.
Genotyping for UGT1A128 is now standard practice before initiating irinotecan. Identifying these high-risk 28A128 patients often leads oncologists to proactively reduce the starting dose of irinotecan, typically by 30% or sometimes more, to mitigate the severe toxicity risk right from the beginning.
Right. Finally, we absolutely cannot limit this discussion just to metabolic enzymes. We have to consider the gatekeepers, the drug transporters. How do defects in transporters, like the example of OATP1B1, affect drug exposure and risk?
Transporters are absolutely crucial. They control the movement of drugs all around the body, absorption from the gut, uptake into target organs like the liver or brain, and excretion via the kidneys or bile. They often determine the overall rate of clearance just as much as metabolism does.
And the key example is OATP1B1.
The most clinically relevant example right now is probably the hepatic uptake transporter OATP1B1, which is encoded by the SLCO1B1 gene. This protein sits on the surface of liver cells and actively transports various drugs, most notably statins like simvastatin, for the bloodstream into the liver cells. That's where statins primarily do their work lowering cholesterol, and it's also where they get metabolized.
And the reduced function variants in SLCO1B1 mean the statin can't get into the liver effectively? Is that the problem?
That's exactly the problem. Reduced function variants, such as the common SLCO1B15, defined by the SNP C.521TC, severely impair this uptake process. When the drug can't efficiently enter the liver cells where it's supposed to act and be cleared,
It stays in the blood.
it remains trapped, essentially, in the systemic circulation. This leads to dramatically elevated systemic drug concentrations. For instance, a patient carrying 2 copies of this reduced function 5 allele, the 55 genotype, has, on average, a 4.5-fold higher concentration of simvastatin acid in their blood compared to someone with 2 normal functioning alleles.
4.5 times higher. And that elevated systemic exposure directly causes the major dose-dependent toxicity we worry about with statins, right?
Precisely. It massively increases the risk of the primary toxicity. Myopathy, which ranges from muscle pain and weakness all the way up to the severe, potentially fatal muscle breakdown known as rhabdomyolysis. The risk for myopathy can be increased up to 17-fold in individuals with the 55 genotype taking higher doses of simvastatin.
Wow, 17-fold.
Yeah. And what's really important to note here is the mechanism. A transporter defect phenocopies a metabolic defect in terms of outcome. Both scenarios, poor metabolism or poor uptake, result in elevated systemic drug exposure and increased toxicity risk. But the actual location, the bottleneck, is entirely different. For statins, testing for these key SLCO1B1 variants is now strongly recommended by guidelines to help guide dose selection or choice of statin, specifically to prevent severe muscle damage.
Okay, that clarifies the transporter role perfectly. Now, we need to pivot slightly. For many of these small molecule drugs, pharmacogenomics helps optimize dosing or avoid toxicity. But you mentioned earlier that for the most advanced therapeutics, biologics, cell and gene therapies, molecular testing often moves from being helpful to being absolutely mandatory. It becomes the non-negotiable prerequisite for treatment eligibility itself.
That's a critical shift in perspective. We move beyond just optimizing a dose by, say, 25 milligrams. Here, the genetic or molecular test determines if the patient gets the drug at all. It's often an eligibility test. The sources we looked at define 3 key strategic pillars for these companion diagnostics that guide the use of advanced biologics.
Right. Let's walk through them. Pillar 1 seems to be focused squarely on safety screening.
Pillar 1 is designed purely to protect the patient from potentially catastrophic adverse events. The absolute prime example, the one everyone learns, is the mandatory testing for the germline allele HLA-B5701 before initiating the antiretroviral drug abacavir, which is used for HIV treatment.
Why is that test mandatory?
Because this specific human leukocyte antigen, HLA allele, is incredibly strongly associated with an almost guaranteed severe multi-organ hypersensitivity reaction, HSR, if a patient carrying it takes abacavir. This reaction isn't just unpleasant, it can be fatal.
So a positive test means?
A positive test result is an absolute contraindication. The drug is simply not prescribed, full stop. The test's utility here is pure prevention. It has near 100% negative predictive value, meaning if you test negative for HLA-B5701, you are considered safe from this specific severe reaction. It's a critical safety gate.
Okay, pure safety. Pillar 2 is about efficacy through target identification. The idea here is the patient must actually have the specific molecular bullseye the drug is designed to hit.
Exactly. This is fundamental for most targeted oncology therapies. The classic example is the mandatory testing for HER2 protein overexpression or amplification of the underlying ERBB2 gene in breast and gastric tumors before a patient can receive trastuzumab, brand name Herceptin.
And why is that?
Trastuzumab is a monoclonal antibody. It's engineered to specifically find, bind to, and block the HER2 receptor protein that's sitting on the surface of the cancer cell. If the tumor doesn't overexpress HER2 or amplify the ERBB2 gene, well, there's simply no therapeutic target present for the drug to hit.
So giving the drug would be pointless.
Completely pointless. It would just expose the patient to the cost and potential side effects of the drug with essentially zero chance of benefit. This testing effectively rules in the specific 20% to 30% of breast cancer patients whose tumors do have the target and are therefore likely to respond to this very effective targeted therapy.
Makes sense. Then pillar 3 is resistance assessment. This sounds different. This test isn't looking for the drug's target. It's looking for internal mechanisms within the tumor that would guarantee treatment failure even if the target is present.
Precisely. This usually involves testing for specific somatic mutations acquired by the tumor cells themselves, not inherited. The standard practice example here is testing for activating mutations in the KRAS gene in patients with metastatic colorectal cancer before prescribing anti-EGFR therapies. These are drugs like cetuximab or panitumumab.
And how does KRAS interfere?
Okay, so these drugs, cetuximab and panitumumab, work by blocking the epidermal growth factor receptor, EGFR, which is upstream on the cell surface. This normally shuts down a signaling pathway inside the cell that drives growth. However, the KRAS protein is located downstream of that very same signaling pathway.
Okay.
If the tumor happens to harbor an activating KRAS mutation, which is common in colorectal cancer, that mutation essentially jams the signaling pathway permanently in the on position, regardless of whether the upstream EGFR receptor is being blocked by the drug or not. It completely bypasses the drug's effect.
Right, so the drug just won't work.
It simply will not work. The activating KRAS mutation acts as a powerful negative predictive biomarker. If that mutation is present, the anti-EGFR drug is contraindicated because we know it will be ineffective. Testing prevents futile treatment.
Okay, those 3 pillars, safety screening, target identification, and resistance assessment, really clarify the role of mandatory testing for biologics. Now, moving to the absolute frontier of modern medicine, how do the genomic considerations change when we start talking about cell and gene therapy, or CGT?
Yeah, this distinction is paramount because the type of testing required reflects the fundamental nature of the disease being treated and the therapy itself. For gene replacement therapy, think of treatments like Luxturna for certain inherited retinal dystrophies or Zolgensma for spinal muscular atrophy, SMA. The testing is what we'd call a diagnostic imperative, meaning these revolutionary treatments are specifically designed to correct an inherited monogenic defect. Therefore, the absolute prerequisite for eligibility is confirming a definitive constitutional genetic diagnosis in the patient. The patient must be shown to have biallelic meaning mutations in both copies of non-functional mutations in the specific target gene the therapy aims to replace. For Luxturna, it's the RPE65 gene. For Zolgensma, it's the SMN1 gene. The test identifies the foundational inherited defect that the therapy is designed to fix.
Okay, so that's about the patient's inherent genetic makeup. But the testing for CAR T-cell therapy, chimeric antigen receptor T-cell therapy, is entirely different, isn't it? Even though it also involves genetic engineering.
Completely different focus. Correct. CAR T-cell therapy is a type of immunotherapy. The patient's own T-cells are extracted, genetically engineered outside the body, ex vivo, to express a receptor that targets a specific antigen on their cancer cells, and then infused back into the patient to hunt down the cancer.
So the testing isn't about the patient's inherited genes.
No. The prerequisite testing here is molecular pathology, focused entirely on the tumor, not on the patient's constitutional germline DNA. Eligibility requires confirming that the patient's specific cancer cells actually express the required antigen target that the engineered CAR-T cells are designed to recognize and attack.
Like CD19.
Exactly. Like CD19 for many B-cell lymphomas and leukemias treated with drugs like Kymriah or Yescarta, or BCMA for multiple myeloma treated with Abecma. The test confirms a characteristic of the disease, the presence of the tumor antigen, which is essential for the engineered T cells to find, lock onto, and destroy the cancer cells. So it's a critical distinction, identifying an inherited defect in the patient versus confirming an acquired somatic molecular target on the tumor. Both are central to advanced diagnostics, but they test different things for different therapeutic strategies.
That distinction is really clear and important. Okay, so we have this incredibly powerful and predictive knowledge, whether it's predicting statin toxicity based on SLCO1B1, guiding irinotecan dosing with UGT1A128, or confirming eligibility for a life-saving therapy like HAR-RT based on CD19 expression. This should be routine practice in every clinic, shouldn't it? But we know adoption is consistently, sometimes frustratingly, slow. Why? The sources mention that every single test must pass something called the translational gauntlet. What is that?
The translational gauntlet is a great term. It represents the rigorous evidence-based pathway that any new diagnostic test, especially a genetic one, must navigate successfully before it can be widely integrated into clinical practice and reimbursed by healthcare systems. Just because a test is scientifically interesting or even analytically accurate doesn't automatically mean it's clinically necessary or beneficial.
Right.
This whole assessment process is often formalized by something called the ACCE framework. It requires 4 distinct components to be rigorously defined, evaluated, and demonstrated for any given test.
Okay, let's define those 4 components. Starting with the 2 that seem to assess the quality and predictive power of the test itself: analytical validity and clinical validity.
Right. Analytical validity addresses the fundamental laboratory performance. It asks the basic question: how accurately and reliably does the laboratory assay actually measure the genetic marker it's supposed to measure? Here, we're focused entirely on the lab's ability to minimize errors, looking at the assay's sensitivity, its specificity, its reproducibility day-to-day, lab to lab. If the lab gets the genotype wrong, everything downstream is obviously incorrect.
Okay, so that's about lab quality. Then C, clinical validity. C, clinical validity assesses the strength of the association between the genetic marker and the clinical outcome. How reliably and strongly does the specific genotype predict the defined phenotype, like poor metabolizer status, or the clinical outcome of interest, like increased risk of toxicity or likelihood of response? This is where we ensure there's a robust, reproducible link, like the near-perfect association we see between carrying the HLA B5701 genotype and the risk of abacavir hypersensitivity. Generally, this component tends to be quite strong for many pharmacogenomic tests because the molecular links between gene function and drug handling are often direct and well understood.
Okay, so those first 2, analytical and clinical validity, seem achievable for many tests. Now, for the 2 components that apparently create the real bottlenecks in implementation, starting with what you called the most critical economic and clinical hurdle: clinical utility. Yes, see clinical utility. This is, without a doubt, the single largest barrier facing the widespread adoption of pharmacogenomics today. Clinical utility moves beyond just prediction. It requires us to answer the crucial question: does acting on the test result actually lead to a measurably improved health outcome for the patient, an outcome that is demonstrably superior to the current standard of care, which is usually empirical dosing or standard clinical monitoring?
So just predicting risk isn't enough.
It's absolutely not enough for broad adoption and reimbursement. You must prove, ideally through large, expensive, prospective randomized controlled trials, RCTs, that your genotype-guided intervention, whether that's adjusting the dose, switching the drug, or selecting a therapy, actually reduces adverse drug events, shortens hospital stays, improves quality of life, or even improves survival outcomes better than just following the standard clinical practice without the genetic information.
And that's hard to prove.
It's extraordinarily difficult, time-consuming, and expensive to prove definitively across diverse patient populations and healthcare settings. Many promising pharmacogenomic tests, despite having strong analytical and clinical validity, falter at this clinical utility stage. The trials often struggle with things like proper patient stratification, choosing clinically meaningful endpoints, having long enough follow-up times to capture the real long-term benefits of avoiding side effects, or simply recruiting enough patients. If robust, positive clinical utility data from well-designed RCTs is missing, then widespread clinical adoption and, critically, reimbursement from third-party payers like insurance companies or national health services simply will not happen. That's the bottleneck.
That makes the challenge very clear. And finally, the essential safety net that determines if the test can be ethically and responsibly implemented, even if it passes the other hurdles: ethical, legal, and social implications, or ELSI. What are the key societal challenges here that must be continually addressed?
ELSI considerations are absolutely profound, maybe even more so for genetic information than other medical data, because genetic data is uniquely sensitive. It's inherently personal, it's permanent, it doesn't change over your lifetime, and it has implications not just for the individual, but also for their biological relatives.
Right.
The primary societal fear, the one that comes up constantly, is genetic discrimination. People worry, quite legitimately sometimes, that their genetic results, perhaps showing predispositions to certain diseases or adverse drug reactions, could be misused. Could it be weaponized by employers to deny jobs or promotions? Could certain types of insurers, like life, disability, or long-term care insurers, use it to set prohibitively high premiums or deny coverage altogether?
Are there protections?
In the United States, legislation like GINA, the Genetic Information Non-Discrimination Act of 2008, provides significant protection against genetic discrimination in health insurance and employment. However, GINA's protections are not comprehensive. They don't currently extend to life insurance, disability insurance, or long-term care insurance, which remains a major concern for many people considering genetic testing.
What else falls under ELSI?
We also face severe challenges regarding privacy and confidentiality. Because genetic data is so uniquely identifiable and immutable, we need extremely robust data security measures and clear policies on data sharing and access. Then there's the huge issue of informed consent complexity. It's genuinely difficult for clinicians to adequately explain the nuances of these tests, their limitations, the probabilistic nature of risk prediction, what the results mean and don't mean, and crucially, they must discuss the potential for incidental findings.
Meaning finding things you weren't looking for.
Exactly. You might do a pharmacogenomic test to guide antidepressant dosing, but the same test might incidentally reveal a high-risk variant for early-onset Alzheimer's disease or a hereditary cancer syndrome that the patient wasn't prepared to learn about. Managing these incidental findings responsibly, ethically, and with appropriate support is a massive, ongoing challenge. Ensuring true informed consent in the face of this complexity is critical, but very hard to do well consistently.
So, given all these hurdles, analytical, clinical, utility, ethical, metany, the regulatory bodies like the FDA and EMA play a key role in trying to enforce some standardization and guide the flow of this evidence into actual practice.
Exactly. The FDA in the US and the EMA in Europe act as crucial arbiters of the evidence. Their most direct impact on day-to-day clinical practice is usually felt through updates to drug labeling. Based on the strength of the accumulated evidence, particularly clinical validity and increasingly clinical utility data, the FDA might update a drug's official label to include specific pharmacogenomic information.
What might that look like?
It can range quite widely. It might be a simple informational statement in the clinical pharmacology section. It could be a cautionary statement in the warnings and precautions section, suggesting that genotyping might be considered for certain patients. Or, at the highest level of evidence and concern, it could be a recommendation or even a mandatory requirement, often highlighted in a boxed warning, stating that an FDA-approved companion diagnostic test must be performed before the drug can be prescribed. We saw this mandatory requirement for abacavir HLA B5701 and for trastuzumab 2 testing. This regulatory action essentially codifies the evidence base for clinical use and can powerfully drive adoption, at least where the evidence is overwhelmingly clear and the risk-benefit calculation demands it.
This has been a truly powerful deep dive into the intricate world of applied pharmacogenomics. We journeyed right from the molecular blueprint of the central dogma, tackled the inherent complexity of genetic variation and haplotypes using concepts like linkage disequilibrium and the star allele system, and saw how sophisticated bioinformatics tools like DBSMP give us population context, UniProt links genes to protein function, and PDB reveals crucial mechanistic insights, like that surprising allosteric effect of the CYP2C93 variant.
We then saw how that foundational knowledge directly translates into predicting patient outcomes, exploring those critical inverted risk profiles of poor metabolizers versus ultra-rapid metabolizers, depending on whether the drug is active or a prodrug. And we recognize the crucial shift where molecular testing becomes absolutely mandatory, not just optional, for guiding the use of the most advanced therapies based on safety, target identification, or resistance assessment.
And finally, we wrapped up by confronting the reality that all this vast scientific knowledge must successfully navigate that rigorous ACCE framework gauntlet to prove its real-world value. We acknowledge that clinical utility proven improved patient outcomes, along with managing the crucial ELSI considerations, remain the largest, most persistent barriers preventing this technology from becoming routine, equitable clinical practice everywhere. The conclusion seems inescapable. The future of therapeutics is absolutely and inextricably linked to evidence-based molecular diagnostics.
It truly is. Which leads us to this final, perhaps provocative thought for you, our listeners, to mull over. We know the cost of sequencing DNA continues to plummet. Generating accurate genetic data is rapidly becoming almost trivial from a technical and cost standpoint. So if the underlying scientific discoveries are proven, if the analytical validity of the test is high, and if the clinical validity, the link between genotype and phenotype, is strong, why is clinical utility, that second C in ACCE, still the largest and most persistent barrier to widespread equitable pharmacogenomics adoption? What specific fundamental shift needs to occur? Is it in healthcare economics and reimbursement models? Is it in regulatory standards for evidence? Or is it in the practicalities of clinical workflow integration? What needs to change to finally close that gap and make truly personalized prescribing the everyday norm, not the exception?
This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.